students, myself Om Prakash Katte and you are welcome to Biology Reward. Today we are discussing class 12th chapter Molecular Basis of Inheritance, and our topic is Hershey and Chase experiment, or this is also known as Blender's experiment. After the grip experiment, the grip proved that the DNA is genetic material and biochemical nature is explained by the three scientists, and they shown that the DNA is genetic material. Now same other experiment given by the Hershey and Chase, and this is called the Blender experiment. By this experiment also it is proved that the genetic material is DNA. So this is another experiment to show that the genetic material is DNA. So for this experiment, Hershey and Chase, they explained some viruses infect the bacteria. So there are the group of viruses which feeds on the bacteria. They kills the bacteria, they digest the bacteria. So the viruses that infect the bacteria, these are called as bacteriophages. So what are bacteriophages? These are the viruses which feed on bacteria. So these bacteriophages are the viruses. So viruses infect the bacteria are called the bacteriophages. They attaches this to bacteria. It means the bacteriophage, this viruses bacteriophage, they attack or infected to the bacteria. And what happened? It is genetic material transferred to bacteria. When the bacteriophage, they attack on the bacteria, so they found that genetic material of this bacteriophage, is transferred to bacteria. And they work to discover. These two scientists, they work to discover what transferring material is, whether protein or DNA. So by this experiment, they both are working or finding out that whether the genetic material is protein or DNA. So for this experiment, they have to observe, they have to find out the genetic material which is transferred, whether it is protein or DNA. So for this experiment, they grew some bacteria or bacteriophages. They grew some bacteriophage medium containing the radioactive phosphorus. So first one, for this experiment, they have taken some bacteriophage, and this bacteriophage, they grown in the radioactive phosphorus. It is P32. It is phosphorus is radioactive phosphorus. So they grew some bacteriophage in medium containing radioactive phosphorus. In other experiment, they grew some bacteriophage in radioactive sulfur. Okay. So in one experiment, they grown some bacteriophage in the medium containing the radioactive phosphorus, and other experiment they grown some bacteria in the medium containing radioactive sulfur. When they grown these bacteriophage, what observed? The bacteriophage lined with P32. So this radioactive substance come in the bacteriophage. In other medium there was radioactive sulfur, so bacteriophage contain sulfur radioactive substance. So here you will see when bacteriophage grown in the, simply you understand, when bacteriophage grown in the phosphorus, So phosphorus come in the bacteriophage. When bacteriophage grown in the radioactive sulfur medium, so this sulfur come in the bacteriophage. Bacteriophage phosphorus is the important component of DNA. So in DNA the bacteriophage come. Bacteriophage S35 sulfur is the component of protein. So the sulfur come in the protein. So by this one, what it is confirmed? It is confirmed that if bacteriophage grown in the medium phosphorus, so it's come in the bacteriophage. So this bacteriophage P32 radioactive phosphorus level, it means the phosphorus come in the bacteriophage. Another case, when the bacteriophage grown in the sulfur medium, so this sulfur come in the bacteriophage. So it is called bacteriophage S35 level bacteriophage. It means when the bacteriophage grown in the sulfur medium, the sulfur come in the bacteriophage. When bacteriophage grow in the phosphorus medium, radioactive phosphorus, so this come in the bacteriophage. Now, there are the three steps in blender experiment or Hershey and Chase experiment. First step is called the infection. First step is infection. Infection is what? This bacteriophage, which is content P32, attack on the bacteria. So infection of bacteriophage on the bacteria. Similarly, the bacteriophage which contain sulfur 35, it attack on the bacteria. Okay, so bacteriophage infection is what? Both bacteriophage attach to the E. coli bacteria separately. So what is infection? This bacteriophage attack on the bacteria. This bacteriophage also attack on the bacteria. So this process is called the infection. Other process is called the blending. Blending, which means mixed up. So genetic material of this bacteriophage mixed with the genetic material of bacteria. What is blending? Blending is a mixing. So bacteriophage DNA is mixed with bacterial DNA. So this is called the blending. So infection, when bacteriophage attack on this bacteria, the genetic material of this bacteriophage mixed with the bacteria, and this is called blending. Similarly, this bacteriophage when attack on the bacteria, the genetic material of both it mixed up, and that is called the blending. Generally, when bacteria are attacked by the bacteriophage, because bacteriophage is the virus, so this bacteriophage, it start digestion of the bacterial DNA, and they produce the large number of virus particles. Now what happened? During the bending, bacteriophage DNA is mixed with the bacterial DNA. So this is second process is called blending. After the blending, third process is centrifugation. When genetic material of both is mixed up, bacteriophage genetic material and bacterial material. Here also bacteriophage genetic material mixed with the bacterial material. Now what happened? Centrifugation. In centrifugation what happened? This is both DNA material and whatever material is there, it is grind up, and by the spinning method, centrifugation method, it means it is rotated. And once it is rotated due to the centrifugation method, the heavy material they settle down at the bottom, and the lighter material they floats on the surface. So this process is called centrifugation. What happened here? Bacteriophage particles separated from bacteria by centrifugation. So by the centrifugation process, bacteriophage particle they are separated from the bacteria by the centrifugation. So when centrifugation is done, after the centrifugation, what happens? This material, bacterial and bacteriophage material, they are separated. So what it is observed? The radioactive phosphorus detected in the same. So this material which was present in the bacteriophage P32, it was present in the radioactive phosphorus detected in the cell. It means this radioactive phosphorus was present in the DNA. So in DNA content there was the phosphorus, but no radioactive in the suspension. So suspension part is the light part, there was the protein. So in suspension part there was no phosphorus. It means the DNA contained phosphorus, radioactive phosphorus. It's proved that the DNA phosphorus is a component of DNA. So this phosphorus come in the DNA, and it is so that the DNA is a genetic material. But no radioactive material is found in the supernated, and supernated part is the protein. So protein, it is not part of the protein. So phosphorus only come in the DNA pellets. But in case of second one, in this one, sulfur. Here radioactive sulfur detected in the supernated. It means sulfur come in the protein. Sulfur come in the protein. But this sulfur not detected in the DNA. It means the phosphorus is the component of the DNA. So it's come in the DNA. And sulfur is a component of protein. So it's come in the protein. So by this experiment, what's conclusion they said? Conclusion is bacteria when infected with the bacteriophage, bacteria infected with the bacteriophage P32 radioactive phosphorus contains radioactive DNA. It means the DNA is having the phosphorus 32 but no radioactive in the protein. So here DNA is get transferred. Second, bacteria infected with the bacteriophage sulfur contains, so this bacteriophage infected to bacteria. So what happened? In such cases, radioactive proteins is observed but no radioactive in DNA. It means in DNA there was no sulfur. Sulfur was present only in the protein, and it indicates that protein did not enter the bacteria from bacteriophage. So only DNA is entered. Protein is not entered. Only DNA is entered from the one bacteriophage to bacteria, because this phosphorus is detected in DNA. In this DNA there was no sulfur, so it's proved that the DNA is the genetic material. So overall in this one we should remember Hershey and Chase gave the experiment. It is commonly known as blender experiment. In this experiment some viruses which infect the bacteria or digest the bacteria, they are called bacteriophage. So when bacteria are grown in the sulfur medium, okay, so the sulfur S35 is come in bacteria. And second, some bacteriophage are grown in the phosphorus medium. So phosphorus come in the bacteria. Now bacteriophage P32, it's infect on the bacteria, and then it is called infection. Then genetic material. Thus in the Hershey and Chase experiment, when bacteriophage, they grown in the medium phosphorus, the phosphorus come in bacteria. This bacteriophage attack on the bacteria, that is called infection process. Then genetic material of both, they get mixed up, it is called the blending, and after the blending the viral bacteriophage material is separated from the bacterial material, and it is observed that the radioactive phosphorus present in the DNA. But radioactive substance phosphorus was not present in the supernated, because supernated there was the protein. So here it is said that the phosphorus come in the DNA, it means DNA is a genetic material. Second case is the bacteriophage S32, when it is grown in the medium, then bacteriophage grown in the medium sulfur, then the sulfur labeled bacteriophage are formed. This bacteriophage attack on the bacteria and genetic material of this bacteriophage, it's mixed with the genetic material of bacteria and then blending occur. After the blending, when centrifugation process, separation is done, so what is observed? So radioactive sulfur was present in the protein part or supernated solution, but this sulfur was not present in the DNA. So it is confirmed that the DNA is a genetic material. Thank you.